Meiosis is a biological process by which a diploid eukaryotic cell divides to form 4 haploid gamete cells. Meiosis is essential for sexual reproduction and therefore occurs in all eukaryotes. Meiosis is defined by 3 laws discovered by Gregor Mendel. The law of segregation states that when gametes form alleles are separated so that each gamete carries only one allele for each gene. The law of independent assortment states that the segregation of alleles for one gene occurs independently to that of any other gene. The principle of dominance states that recessive alleles will be masked by dominant alleles. Before meiosis starts, the cell is in interphase. During interphase, the chromatin is replicated. The chromatin is uncondensed, and the cell makes 2 centrosomes. After the interphase comes meiosis, a process that is broken up into 8 basic subphases. First is prophase 1, the longest stage of meiosis. During the phase, the chromatin begins to condense into visible chromosomes, and the homologous chromosomes start to loosely pair with each other in a process called synapsis. The synapsis allows the homologous chromosomes to undergo crossing over, in which chromatids exchange alleles with another. These homologous chromosomes pairs form tetrads that each have chiasmata, where crossing over has occurred. Furthermore, in this phase, the nucleoli disperse and the centrosomes form microtubules that are made of spindle fibers that move towards the centrosomes and attach to kinetochores. The spindle fibers begin moving the homologous chromosomes to the metaphase plate.
Yeah, that's what it's no, but then we go like this. What I'm thinking is
Next is metaphase 1. In this phase, the homologous chromosomes / tetrads line up at the metaphase plate with one chromosome in each pair facing each pole. Both chromatids of one homolog are attached to kinetochore microtubules from one pole. Next is anaphase 1. In this subphase, the spindle apparatus separates the homologous chromosomes, moving each one to the opposite poles. However, the sister chromatids are still held together through cohesion at the centromere. Next is telophase 1 and cytokinesis. When this phase begins, each pole of the cell has complete haploid set of duplicated chromosomes. In animal cells, a cleavage furrow forms, and in plant cells, a cell plate forms, allowing cytokinesis to occur simultaneously to telophase 1. This phase forms 2 haploid daughter cells. After this, the cell continues to meiosis 2 without chromosome replication. Next is prophase 2, similar to prophase 1. In each cell, the centrioles form a spindle apparatus. The spindle apparatus helps the sister chromatids move towards the metaphase 2 plate. Next is metaphase 2. In this subphase, the non-genetically identical chromatids, because of crossing over, are positioned at the metaphase plate. The kinetochores on each chromatid attach to microtubules on opposite ends.
The last stage is telophase 2 and cytokinesis. The nuclei.
Wait, you need to get the nucleoli on both cells.
I said nuclei, not nuclei.
Oh, wait. Just a nucleoli.
Next is anaphase 2. In this subphase, the centromere that was once holding the sister chromatids together is separated, and the spindle apparatus moves the chromatids to opposite poles.
So no nucleoli?
Finally, the last subphase is telophase 2 and cytokinesis. In this subphase, the cells split and 4 haploid daughter cells are formed. Each one is genetically distinct. The nuclei of each cell reforms and the chromosomes begin to decondense. In males, through a process known as spermatogenesis, each haploid daughter cell becomes sperm cells, creating 4 sperm cells in total.
Are we dividing it? Are we putting it together and then dividing it? Because how are we gonna? Are we putting the nucleus in the nucleus and then we're dividing it? Do we put the nucleus on first and then it divides, or nucleus